These are the steps of mitosis. The first step of mitosis is interphase. Interphase is the first phase before division, or DNA copies itself. The DNA is in the nucleus, but the spindle fibers are not present. The next stage of mitosis is prophase. Prophase is when the sister chromatids, their identical copies, line up next to each other. The DNA is paired in chromatids, but the spindle fibers are still not present. Phase is when you line them up in the middle. Is when you line all of the sister chromatids up in the middle. Anaphase is when the sister chromatids are parted and are moved to different sides. Telophase/cytokinesis, when they've reached the ends. Sister chromatids have reached their ends. What anaphase looks like. This is interphase.
And the spindle fibers move to other sides.
The sister chromatids.
The next step is telophase. I love speaking now.
This is metaphase.
The telophase is when the sister chromatids finally get to the end.
As you can see,
And
the sister chromatids are lined up in the middle.
so the new nucleus starts to begin making itself.
This is anaphase. This is what anaphase looks like.
This is when the cell splits and the new nucleus.
These are no longer paired together. They are now split apart by the spindle fibers.
And the spindle fibers still are not there. They are going down.
See, they're split at the different ends. Telophase. In telophase, the cell is starting to split. So all of the sister chromatids are still at their sides.
Let me rephrase.
That is telophase.
The final cells, the two final cells.
Meiosis is when, at the end, there are two nucleotides at both ends of the cell, and that is cytokinesis. So those are the steps of mitosis. But let's move on. These are the steps of meiosis. In meiosis, there are normally two of the mitosis steps. Here we go. The first stage is interphase. This is also the stage before division. And this is what this one looks like. The second stage, which is called prophase 1. Prophase 1 is when all of the homologous chromosomes, or sisters, pair up all over the cell. There are always two of these in meiosis at prophase 1. The next phase is metaphase 1. That's when the homologous chromosomes, or the sisters, line up in the middle. But there are a lot of them. And they're doubled. And they're double. And there are 4 chromatids wide. So it's actually pretty cool because it's double than mitosis. The next stage is called anaphase 1. That's when the the homologous chromatids or the sisters are pulled apart, but there's still one of the homologous chromatids because they were doubled as they were pulled apart. That made no sense. Since in the last one they're all doubled. So since they're doubled, they're all paired up, so they're pulled apart.
So basically, if you can work with those, so there's
One of the last phases is telophase 1 and cytokinesis. This is telophase 1. Telophase 1 is when the cell starts to split. The first steps of meiosis are a lot like mitosis. So the cell splits. And there are still sister chromatids. So that is telophase. And then cytokinesis 1 is when there are both two. Okay. Forget that I said that. Never mind. So the next step is prophase 2, and this is when we start doubling up, and it is not like mitosis anymore. Prophase 1 is when the sister chromatids pair up in both of the cells.
But then the new cells, which will
when they all pair up in the different cells, which is pretty crazy because there are two cells and there are sister chromatids in it, and the nucleus is not yet formed. Yeah. Okay, so yeah. So metaphase 2 is when the sister chromatids line up in the middle of both of the cells, which is kind of crazy, but they have to follow the same systems.
well, they are actually similar to the interphase.
Anaphase 2 is when they are both and they're pulled apart at the different sides of the cells.
That is meiosis.
Telophase 2 and cytokinesis 2 is when they start to split, kind of, and they're all there, and they're the nucleus, but there are many cells made if you use meiosis. There. So those are the meiosis steps. The meiosis steps are basically doubled from the mitosis steps. If you reproduce via mitosis, this reproduction system is asexual. When you produce via meiosis and fertilization, this form of reproduction is sexual. Some environments that favor these forms of reproduction, which are asexual and sexual. Some environmental conditions where it best succeeds with sexual is complex species, changing environments, busy environments, which is kind of like competition, predators, or diseases going around. But some environmental conditions where it best succeeds for asexual reproduction are simple species, and then some of these simple species are bacteria or single-celled organisms, and then some other environmental conditions for asexual are harsh environments, unchanging environments, kind of, yeah. And then advantages of sexual reproduction allows variation, like adaptation or evolution, and then two parents, which make it easier to protect all of the babies. For asexual reproduction, advantages of this method are more kids and less time. Some animals that use that use asexual reproduction are grizzly bears, and other ones are sand scorpions. But blue-headed grays also use sexual reproduction. But some animals that use asexual reproduction are bacteria. Starfish also use asexual reproduction. What is next?
That is the end of Taylor's Top 8.
Made with Doodlecast Pro.